Good evening, everyone. Thank you for joining us on this third Armin Labs and AONM webinar on COVID-19. We'll start in just a minute or so.
Hello, everybody.
I think I'll just begin introducing our distinguished speakers because there's a lot to cover this evening. Thank you again, everyone, for joining us today. This is the Academy of Nutritional Medicine with Dr. Armin Schwarzbach and Professor Leona Gilbert from Finland. I'll introduce Professor Leona Gilbert first because she's going to be speaking first. She's an adjunct professor of cell and molecular biology at the University of Jyväskylä, Finland, and the CEO of Tested Oy. She has a doctorate in biotechnology and specializes in bioinnovation and biobusiness. Her numerous peer-reviewed articles span research results that she turns into translational value with developing novel delivery vectors that can be used for imaging and biotechnological tools, as well as developing diagnostic lab-on-chip platforms. Her research initiatives into complete diagnostic platforms and clinical profiling of patients for tick-borne diseases allow a better understanding of how chronic conditions can be established with tick-borne pathogen infections. And Dr. Armin Schwarzbach, who's a regular guest with us in our webinars as well as our conferences, will be familiar to you all, I'm sure. He's the founder and medical director at Armin Labs, Augsburg, and co-founder and former CEO of the Borreliosis Clinic in Augsburg, Germany. He's a medical doctor and specialist in laboratory medicine with over 3 decades of experience in clinical medicine and laboratory testing. As well as being a clinician for many years, he's tested over 70,000 patients for Lyme disease and co-infections, and is now, as you know, conducting tests for SARS-Cov-2, the causative agent of COVID-19 as well. So without further ado, Leona, I'd like to turn the floor over to you, please.
Okay, let's see if this works. I think somebody is trying to ask a question as well. If somebody could actually organize that, that would be great. I'm going to...
are we able to write your name? I'll write your name on a chat box while you're speaking, Leona. Yep.
Sounds good. Okay, I'm going to share my screen with you, and I hope everybody can see it. So, hi, and thank you for that welcome, and thank you for that bio as well. So I am the CEO of Tested. It is a university spin-off company, as Jillian mentioned, and we have developed a complete SARS-CoV-2 serological test, and I'd like to spend the next minutes talking to you about this. And let's see if it works. Yes. Okay, so Tested mission is to provide appropriate testing for COVID-19 disease patients so that the patient can be treated, recover quicker, and return to normalcy, return back to work, return back to loved ones. And this is really our goal or our mantra in our company at the moment. As we know, SARS-CoV-2 is pandemic. As of today, we have 3.7 million confirmed cases all over the world. And unfortunately, the deaths are increasing daily as well. And due to this crisis, this global crisis, Tested really is committed to try to improve the situation, not only here in Finland, but also globally. So in doing so, we have developed this SARSPLEX. SARSPLEX is a SARS-CoV-2 antibody test. And it is CE and IVD marked, which means that in Europe, it is conforming with the European guidelines and the European harmonized standards in producing a diagnostic test of this type. So because it is CE and IVD marked, we're able to market it and sell it in Europe and also Canada recognizes CE and IVD as well as Australia, and of course Mexico and China, and FDA is getting there as well. Not only is this antibody test actually conforming with guidelines and harmonized standards for efficacy, safety, reproducibility, but it is also manufactured in ISO 13485 accreditation laboratory that's 2 meters away from me. And because it's manufactured in this quality management system, again, national authorities like FDA, like Health Canada, they recognize this standard. So it's really this product is produced in a quality management kind of system that allows reproducibility and validation that this test kit is safe, it's reproducible, and is doing as it should. So what is SARSPLEX? SARSPLEX is an ELISA-based platform. So you can see this ELISA plate on the left-hand side of your screen. And SARSPLEX is testing 3 different types of antibody responses against 2 very specific proteins from this novel 2019 SARS-CoV-2 coronavirus. And because of this testing 3 different antibodies and testing against 2 different proteins, we are calling it this multiplex system. So Tested as a company have a philosophy to multiplex these diagnostic test kits so that you don't need to have 6 tests being performed to get the same information as we're providing. So as I mentioned already, SARSPLEX is CE and IVD marked, okay? And it is manufactured once again in this accredited ISO 13485 facility that we have here in Jyväskylä, Finland. Now SARSPLEX is really a novel kit for this novel coronavirus because we have multiplexed it. We have incorporated both the spike protein from the virus, the S1 protein, and the nuclear capsid protein to enhance the sensitivity, specificity, and the overall robustness of this diagnostic test kits. A lot of the test kits out right now that are being utilized usually incorporates 1 protein, not 2, and a lot of the test kits that are available now are testing 1 or 2 antibody types, and we here are testing all 3 antibody types, IgM, IgG, and IgA, against these 2 different proteins. So in a sense, this multiplexing platform puts 6 different tests onto 1. And it's important to use all 3 antibodies because it can demonstrate seroconversion and immunity in these patients, as others have seen as well. So I'd like to spend the next few minutes in actually discussing the validation study that we did in order to not only prove that SARSPLEX is functioning as it does, but also to validate the results that we get from this diagnostic kit. So we did a clinical study where we pooled 149 sera samples and we tested them against our 2 proteins, and we tested for the 3 antibodies. Within the sera samples, of course, we have characterized SARS-CoV-2 sera specimens. We have actual patients that have Epstein-Barr infection. We also have patient sera that has a mycoplasma pneumonia infection. We also have sera from those individuals that have a human parvovirus infection. We have also individuals from Finland that have experienced summer flu from 2018. And of course, we have healthy sera samples or pre-2019 coronavirus specimens from 2016. Now, why did we actually include Epstein-Barr, mycoplasma, and human parvovirus, of course, summer flu? Because we wanted to show if there would be any cross-reactivity of these patients against our proteins that we're using within this test kit. And we'll show you the data that we obtained from these individual sera samples. And also, the validation occurred because we had de-identified, disregarded, and donated samples that were actually donated from healthcare units here in Finland and other places according to their kind of accreditation standard. And they were able to donate these samples for research purposes according to principles set forth in the Declaration of Helsinki and our Finnish medical act and law. We also purchased reference samples, okay, to make sure that this test kit is functioning properly from Discovery Life Sciences. So once again, here is the list of the type of samples that we have in our study. So SARS, Epstein, mycoplasma, human parvovirus, summer flu, and healthy samples. So, and now I would like to get into the results. So what you see here first is actually the IgM immune response against the S1 and the N proteins of this coronavirus. You can see if we start from the left, the individual group samples are the SARS positive samples here, or SARS characterized samples. And then we have, of course, Epstein, then mycoplasma, human parvovirus. More importantly, we have the pre-2019 samples, and we have, of course, have the summer flu samples. And you can see here that we have these dotted lines corresponding to the cutoff mark and the border lines dictated by actually the positive samples in SARSPLEX and the negative samples that we see actually in healthy individuals or summer flu individuals. And what's important here in the take-home message is by looking at actually our summer flu and our pre-2019 coronavirus samples, you can see that there is really no cross-reactivity, okay, or unspecific binding to our antigens. But interesting enough, we have Epstein-Barr factor, actual SARAs responding positively, and yet they're not. And this is actually quite characteristic with IgM antibody. There could be this sense of unspecific binding in some cases. And this is just quite typical of IgM immune responses, that we will see some unspecific binding. And this is something to look at when we're actually dealing with these type of individual patients if they are suffering from Epstein. They may have a false positive response here in this test. But this is not exceptional to SARSPLEX. This is seen in other antibody type testing as well. But if we now move on to IgG response, so IgG response is interesting as well. So we have a cutoff mark in the borderline here, and we're taking very cautiously looking at the summer flu and the pre-2019 coronavirus samples and seeing if they actually match well to the negative coronavirus specimens that we have had. And they actually correspond quite nicely to the negatives are negatives, the positives are positives. Again, we see Epstein, and we have a mycoplasma pneumonia showing false positives here. And again, this is not exceptional to these types of testing in these types of patients. Other people have seen this as well, but these numbers are substantially low. Oops, let's go back here. And it's interesting too, again, with summer flu individuals, and that's traditionally influenza virus patients, you can see that there's no cross-reactivity. And also with pre-2009 coronavirus individuals, the healthy individuals, there is no false positives here. And that's really good at this point as well. So now this is interesting. This is IgA immune response. And IgA is important for us because it usually resides in the mucosal area. So those areas of the digestive tract, those areas of the lung or reproductive tracts. And it's the IgA that is the first responder if it's present to fight off those viral infections within the lungs or those mucosal areas. And I want to take a look here that actually our negative controls, which are the pre-2019 coronavirus samples and summer flu samples, can be seen here on the right-hand side. But if we zoom in the next picture, you have a better representation here. But take a look actually at the characterized coronavirus samples. The positives are positives and the negatives are negatives, as they should be. But again, we have this interesting phenomenon that Epstein-Barr viruses, again, those individuals are showing some positive response here. It is not a lot. It's 0.5 in the optical density reading. So it is still present. So we have to be careful when dealing with those individuals. And again, this is not exceptional with SARSPLEX. This is a phenomenon that we see in a lot of antibody-based testing. And there's a citation that can actually show this unspecific binding in IgA. But what's exciting is, again, I can't stress this enough, that we have with the healthy controls pre-2019 and the summer flu individuals, there is no positive cross-reaction at all. So this is say against the two proteins, the nucleocapsid and the spike 1 protein is performing quite well. Now, if you look at the robustness of the test kit in this diagram here, you can see the sensitivity on the left-hand side and the specificity on the right-hand side. We put them individually here, so 77% sensitive for IgM, 100 for specificity for IgM. For IgG, it's 100 and then 99%. And for IgA alone, it's 83% and 100%. These are if you're going to analyze the robustness of the results that you have on an individual antibody base. But because we are multiplexing, we are putting all the three antibodies together to get a holistic view of the antibody response in that individual. And you can see once we take that information together, we are at 100% sensitive and 100% specific in testing these individuals. And this is important because this multiplexing of these antibodies to get a holistic view is important to see how immune fit the individual is. So I also want to point out another additional study that we did. We wanted to see how our test was responding to the reference test or an index test that's already been used elsewhere. For an example, Eurimmune's test, or for example, Roche's test. How does SARSPLEX compare to that? So we did a positive agreement. So if positive patients are positive in one test, are they positive in SARSPLEX? And then we did a negative agreement. Is it negative in their test and negative in our test? And we got actually some percentages of that. And we have a positive agreement with this reference test at 83% and 100%. And I wanted to mention in the sensitivity, these percentages are not 100% because other test kits only test for one antibody or one protein. And this is what we're comparing our two protein testing for 3 antibodies against a test that only tests for one antibody and one protein. So we're never going to have 100% match here. But we're doing really quite well with the industry standard, and we have a really nice positive agreement and negative agreement within this test. We also then, of course, for the robustness to analyze how accurate it is, we have a false positive rate, okay, in specificity at 7%, false negative rate of 15%, okay, and that's mostly because of the IgM. Again, we do see some unspecific binding. And overall, if we look at the agreement observed according to statistical analysis, when we analyze all 3 antibodies, we almost have perfect arrangement with reference tests. So this positive and negative agreement is really good to do when you're comparing one test against the other, but we have to keep in mind what the tests are testing for. Are they testing for one antigen or 2 antigens? Are they testing for one antibody, 2 antibodies, or 3 antibodies like we have in SARSPLEX? So this is the interpretation of the results, but Dr. Schwarzbach will be actually going into this a little bit more details to really give you an understanding of what this means in a clinical perspective. So I'm just gonna leave this here and move on to the next slide. And I just wanted to point out there is a benefit to actually investigating of serology because we can track ongoing outbreak and we can have retrospective assessment of the attack rate or the extent of the outbreak in a community, in a nation, and as a globe. So it's really beneficial that we actually start testing for serology. Also, other benefits of SARSPLEX, okay? So it's vital for control and resolution of a viral infection, especially SARS-CoV-2 infection, that we analyze the immune response. Both the mucosal IgA response and systemic IgM and IgG immune response will help in fighting a viral infection. And as a virus infects the lungs first, it's important that IgA is within the testing scheme to indicate immune fitness to combat and possibly resolve this viral infection. And this is the reasoning why we put IgM, IgG, and IgA together on one platform for testing. So I want to, at this point, thank you for your attention and entertain any questions that you may have. Thank you.
Thank you very much indeed, Leona. That was very, very interesting indeed. I think a lot of that was new as well for our listeners. And I'll just look and see if there are any questions. Armin, is that all right before we go on to you? I'll just ask a few of the questions of Leona. So Leona, one of the questions that's come up is, does this mean that people with underlying Epstein-Barr are more prone to get infected with COVID-19, in other words, SARS-CoV-2?
I don't know if we can answer that question more prone and have a general statement, but I think that if your immune system is compromised, of course, I would definitely consider that a more high risk than anything. And because I don't know the individual on what their symptoms are, their history, it's hard to say if they're going to be more prone or not. But I can say that we have to pay attention for those underlying infections that might give a false positive result, okay, in any type of antibody diagnostic test. As I said, it's not exceptional just for SARSPLEX.
Okay, thank you. And then another question is regarding the validation test. Who were they tested against?
Who were they tested against? The validation.
Yeah, I'm not sure if it's possible to answer that, but that's how the question is asked. Who were they tested against in the validation? That was presumably the figures that you showed us with the different.
Yeah, I can't say who because we had de-identified the patients and the people, but we had SARS positive and negative individuals. We have Epstein-Barr positive and negative individuals. We had mycoplasma pneumonia individuals as well, human parvovirus, because human parvovirus is my favorite, and summer flu individuals, okay, and also then healthy sera samples pre-2019 coronavirus. If that, I hope that answers the question.
Thank you. Yes, I'm sure she'll come back with a further question if not. And then we're also getting the question, who carries this test in the UK? And that, of course, I can begin to answer at least, because that's the Academy of Nutritional Medicine. And that's Armin Labs in Germany, specifically. And because the Academy of Nutritional Medicine represents Armin Labs in the UK, you can find out more about our tests at AONM.org. And there you'll see that we have a COVID-19 page. It does actually come up on the because of the importance of it. I think if you turn to aonm.org, you'll immediately see that page and can click onto it for further information about both the SARSPLEX tests and the others that Armin has and offers. And that maybe, Armin, let me just scroll down and see. There is another few questions here, Leona. Would you say that the Epstein-Barr that was found is mostly dormant? I know that on Armin's tests he has lytic and latent, lytic being currently replicating and latent not, even though it is in the innate immune system. What would you say there?
Well, I know exactly. It's actually latent. It's latent.
Okay.
Yeah.
Okay, thank you. And might the summer flu vaccine or even the winter flu vaccine interfere with the accuracy of the test result?
Well, actually, I have to say no, because these individuals, summer flu individuals, some of them have had their vaccine. This I know of. So it didn't give a false positive response.
Okay. And for how long does the test detect the COVID-19 IgM antibodies found in the blood?
Yeah, so we have a limit of detection, and it's actually the limit of detection for our test kit is I believe, if I'm right, it's 50 nanograms per milliliter. So if your antibody drops below that, then we will not be able to detect it. But if you're asking the question, how long will IgM stay within the blood? Well, actually, Dr. Schwarzbach is going to actually present a slide in regards to the antibody titers and how they go up and how they go down. So I will wait for that.
Okay, well, let's maybe switch to Dr. Schwarzbach. There are further questions, but we can go into them later. So Dr. Schwarzbach, Armin, maybe you could present, I know you had some specific information on the immunoglobulins.
Yes, hello everybody. I want to show you now the slide, what's now the consequence of the presentation from Professor Gilbert. Let me say two sentences. The SARSPLEX was the first assay in the world before all others testing IgG, IgA, IgM the same time. We had other ELISA before, just for IgG, IgA, and we had quick tests for IgG, IgM. So that's a little history behind that, but congratulations to Professor Gilbert to having developed the first and CE marked and also validated and IVD registered. So we were able to do that, and we have now more and more experience in that. And I want to show you in this slide how our immune system works. This not just for SARS coronavirus, but on the left side you see that it needs time to develop antibodies. What is the first antibody we develop? It's the IgM. IgM antibody is normally, we think, also in the COVID-19 around 2-3 days. So maximum 7 days. So it can give you surely information if your swab is negative. We have problems with the swabs, with the PCR. There are some studies also from China that sensitivity of swabs is around 59% and of NMR is around 69%. So we know the technique is not the best. Some are doing so. We developed the sputum. And today I had one case, interestingly, coming to my laboratory. This was a guy who wanted traveling to Czech Republic. They need a document, a healthy document that they are negative in the PCR. We use the protocol from Professor Drosten. This is the European protocol, and I know also British patients are sending to other laboratories in the beginning. Also NHS sent samples to German laboratories for that protocol, so it's very established. But in the swab, we did it also. It was inhibited as it was, we could not say what it is, but in the sputum, in the deep throat, we found it positive, also in the confirmation test. So that means that it's, as Leona Gilbert said already, that it's in the deep throat better maybe than in the nasopharyngeal part. And there's a tendency, let us see where the data is going there. So nowadays I would say I would prefer more the sputum for this, but there are different opinions about that. It's allowed in Germany to do sputum, to do swabs. Both are allowed, both are fine, but I'm sure the techniques are different. Coming back to the antibodies. The antibodies, in the beginning, you have an IgM, but as Professor Gilbert said, we have also IgA production. IgA, maybe to add to this, it's not just produced in the mucosa, it's also produced systemically. Otherwise, we could not find it in the serum. That means 80%, around 80% produced systemically in the system, and around 30% in the mucosa. We have it also for other mucosal infections like H. pylori, we do IgA, we do it for chlamydia, pneumonia, we do it for chlamydia trachomatis, we do it for Yersinia. We do a lot of IgA testings because this is the only marker we have the chance to find in mucosa infections, inflammation. And this is surely the infection which starts in the mucosa for sure. You all know the virus is starting in your nose, and then it's moving down into your lung, and then you get the problems there maybe where you get bad issues. So the problem is the IgA is coming a little later, or not, it's a problem. IgM first, you see simultaneously maybe IgA, we cannot say exactly. And the IgG, where you all know this means a former infection, it's a past infection, how you name it, it's coming very late, to say that way. Also in COVID infections, we have a lot of experience now with it, and what we can say by a lot of patients we diagnose, characterized patients who are positive after quarantine 2 weeks, that some patients they express IgA antibodies 3 weeks after getting the first symptoms, 3 weeks, and the seroconversion started to IgG IgG antibodies 4 weeks or later. So IgA seems to be also a problem with unspecific reactions for sure, but what I can tell you, we diagnosed a lot of characterized patients with positive swabs, with proven infections, and we find a lot of IgA, not IgG. In the beginning for a while, or maybe the whole persistent infection. We have also now the discussion, maybe to mention that, we have patients in Germany that have PCR positivity for 4 or 6 weeks or longer. They are positive in swab. We think more and more, you saw the study from Korea, that they thought it could be a reactivation, but today I got the message, or you can find it in internet on the website, that it's a dead virus, because PCR cannot distinguish between active virus or healthy virus and dead virus. It's a protein, and so we think in these few cases where you find swabs over months or maybe some weeks, it could be a dead virus detected by PCR, but the patients are completely asymptomatic. Also, the patient coming to my laboratory had the cough in February or March, so it was now, I think, 6-8 weeks ago, but he seems to be still positive by PCR in his sputum, and that's proven case as an example. So we have exceptions from every rule, that's for sure. A normal immune system should produce IgM, IgA, and IgG. You can see it on the left side within a few days, and the persistence of the antibodies, we don't know exactly about that, because we don't have long-term studies, how long IgG could persist, how long IgM could persist. You see the days, 240 days, but you know, the infection, the first case in Wuhan, it was it was in November, December. So we don't have long-term data about that. We need for sure more data in the follow-up, in seroconversion and all, but we get now more and more impression about that. What I can tell you, within 3 weeks, after 3 weeks, starting the first symptoms, 94% of the patient produce antibodies. This is also a new study from coming out now by thousands of patients with the other test system. So we get more and more evidence for that. On the right side, you see how, if we discuss the vaccination and immunization, you see also IgM production, IgG production. In the first phase, the same, IgM is coming 7 to 10 days, whatever, or 3 to 4 or 5 days. It's individual profile we have there. The IgG is coming later. We normally show it after vaccination 3 weeks or 20 days in this case, and then we maybe need a booster. We need a second vaccination to induce enough protein outside. Antibodies are proteins. Maybe also I mentioned IgG can bind one pathogen. IgM has 5 binding arms. It can bind 5 pathogens, and IgA 2. So all are protection, all are natural immune systems. So it's good to have antibodies, not it's bad. The cross reactions Leona mentioned are surely a problem, and the more and more I'm thinking about that, IgM, I think if we find isolated IgM, we should also check better maybe for EBV virus in these cases. Maybe these people have really no COVID, no fresh or no current SARS infection. They have more by maybe an EBV infection or CMV. I don't know data about CMV, Leona. Maybe you could also give us an impression there. I want to show you now a sample report. We are already doing a lot of this with the SARSPLEX, not just for England. We are doing a lot of this SARSPLEX for Germany, for German patients, also for hotspots. Very interesting.
Could you enlarge it a little bit, Armin, because it's very small on our screen.
We have in Germany, you heard about that, we had a Corona strong beer party in Mitterteich, and a lot of patients are infected.
Yes, that's much better.
You can see it now. We had this hotspot, you know, it's a hotspot situation. In Rosenheim is one hotspot, and one was in Heimsberg, where one of our professors is stricken, and the other hotspot was for sure the strong beer party, end of February, beginning of March, and horrible outcome. The whole village was isolated that time in Germany. The people not allowed going outside, and we do a lot of testings now there, and we find a lot of antibodies. I would say 70% are positive in this community, but that doesn't mean that 70% are sick. They are running around asymptomatic, so we have a lot of very high dark number behind that. Today I saw in our news that 10 to 11-fold higher is the dark number than the registered number with positive swabs. It's in Germany 160, 170,000. But if you say you do it 10-fold higher by Robert Koch Institute, that was their information today, then you have 1.5 million Germans already infected. And
do you, sorry, do you have a positive here? This is a negative. Could I just ask, please, what
yes.
ODI means here? Because normally on your tests we have SI for early spots, which is stimulation index.
Yes, Leona, you're the expert, the developer. What is ODI?
It's the optical density index.
Could you explain that, maybe?
Yeah, we're taking the optical density and we're dividing it by the negative or the healthy controls, so that we could have a number of from 0 to 4. So we're putting it in an index. We're normalizing the values so that we can read it from 0 to 4. So 0 means really as the borderline, as Armin can show in the different testing here, that you have negatives, then you have borderline, and then you have positive. So anything above 1 is positive, and then the borderline is there in the negative.
How should we interpret the borderline, Leona? Because I know a lot of people will ask about that. Is that something that you need to retest in a couple of weeks and see whether it was really positive or negative, or well, how would you see that?
I think that actually this is a question for Armin, okay? And I can say what I think, okay? I have to say some clinical laboratories honestly take borderline as weak signal, and some people take borderlines as negative. Okay, and that's the point. That's, I think I would, if I were a clinician, I would survey the patients with their symptoms, okay, and see how they are feeling, because really it's a practice that the clinical laboratories use. Once they go and test like thousands of samples, they can get a feeling of what that borderline is. We set the borderline according to analytical standards and analytical reference points. Okay, we want to make sure that positive is positive and negative is negative without, and disregarding actually, background noise analytically that's coming from SARA, because SARA is very messy when it gives us signal. So that's why we have a borderline when we set this up in such diagnostic test kits. And it is really the clinical laboratory to analyze the borderlines and see, based on their experience with this test and patients or people that they use to test on, to see if they're going to say that that's weak, or that's weak positive, or is that negative.
Thank you. Armin, maybe you have just one positive test to show us as well. Not
yes,
that,
but
it will change much. Yes.
But I want to discuss now, what does it mean if you have these 3 negative antibodies? This is the majority we test in Germany. As you know, not everybody had it. Also, it's not excluding the incubation time. Incubation time is up to 2 weeks. This is really a dangerous time where you can infect others, and you are a silent carrier that time. But on the other side, if you don't have antibodies, I could tell you there was in that day no sign for a contact with the virus, but you could still be in this incubation time. We cannot exclude that by negative antibodies. So the interpretation in this case, if all is negative, no serological indication of an infection with SARS-CoV-2. Symptoms, every medical doctor says symptoms, symptoms, symptoms. And this is extremely difficult, I know, in the differentials, but it's so important to check the symptoms for sure. And everybody, if you have heard about the symptoms and also the exposure. We have done like our co-infection checklist. We did a checklist for that some weeks ago in the beginning, and we said, okay, the exposure, WHO also does a definition of exposure if you went for skiing or if you went to South Tyrolia or wherever you came from, Ischgl back, this generation, or from China if you did business travel. But what I can tell you, the exposure is now more. The first wave is in Germany now over. And we had now, a lot of us had exposure by friends, by neighbors, in communities, working places, employees. So we have more and more exposure now in Germany for sure, because it's now, as Leona said, endemic, epidemic, pandemic, and or hotspot. You have it in some company, we have it, in the other company not. So similar to Lyme disease and other infections. But if you don't have antibodies, you could say there's a very, very high probability, nearly 100%, you didn't have it. Maybe you in this incubation phase, we cannot exclude that. So still you need protection. You need to follow the guidelines of your authorities. You're not allowed to run around with this and say, oh, I had it already. Maybe I have some protection. I have some immunity, who knows? But this is a situation where you could not be so safe as others who already have the antibodies. And I show you now an example of a patient with antibodies, also by SARSPLEX. This is a typical, I need to make it a little, so now you can see that, sorry, little this direction. This is a patient with a positive IgG, and you see it's clearly positive. It's not a borderline. So it's not questionable, this case, that's surely, and I could say you, so this patient is per definition not infectious for others, no infectivity, because the antibodies are produced 3 weeks or let's say 2 weeks after your quarantine time, the IgG. So what I can say you, this patient had it. The question of immunity is a completely different one. How long, what is the period, how long you are protected by that? Maybe it's immunity for 2-3 years, who knows? Today we got some information also about some models about that, but we cannot tell you how long you have a vaccination titer. This is an active vaccination you're getting from a huge virus. It's not a passive, it's not an attenuated virus you get vaccinated with it. But I can tell you in this case, clearly positive IgG antibodies, 2.5 ODI, and this is positive. And then I can tell you this patient had it, and we think that these patients have some kind of immunity for a while. Some say 2 to 3 months, some say 2 to 3 years. Who knows? We don't know it exactly. Also, what we cannot tell you if there's a virus mutation, if there's really protection against that. We have some information from Professor Drosten. He did some samples from the, like Leona did, from corona patients from 2008 it was that time, with SARS that time, and he found that 30% had CD4 positive cellular immune reactions against COVID-19. Also that they react not so strong in the symptoms afterwards. So maybe a part of an immunity against this by your cellular immune system, because we're not talking about cellular immune system now, we are talking about Th2 system, and you know, in every system in the world you have Th1 and Th2 reactions, like we do ELISpots, and I'm pretty sure the ELISpot will come. It's in development to complete the whole picture, to say with who has now cellular immunity and humoral immunity, and who is reacting still on the cellular cellular reactions against this. So there's a lot of science coming now for sure from different sides, immunologists and cellular specialists, and like Leona Gilbert with the antibody testings. So very important is to have the complete picture. I cannot show you now, we don't, the time was a little short to show you some examples, but next time we can show you more. We have also patients with IgG and IgA antibodies, but that means the symptoms were 3 weeks ago. So this patient's out of this. And if you have antibodies, you feel safer than people without antibodies, for sure. This is absolutely sure. Also in companies where we go. I have an example for you. We do also company testings, and when you have a pilot, you have two pilots, the pilot and co-pilot, and one pilot is the A team, the other is the B team. A team, these are the people who had it already without knowing they have IgG antibodies against this, or maybe IgG and IgA together. And the other co-pilot, he has no antibodies. So that means you have still one pilot on a huge plane flying around, and the B team could still be infected and could be in the incubation time. So the B pilot has a high risk that he could breathlessness and that the plane could crash. So, but if you have two pilots from the B team without antibodies, that means a very, very high risk that they could get symptomatically, they can infect each other, they get breathlessness, and the plane will crash. So it's always good to have an A team and B team. I have it in my laboratory also. So that means we have some quarantine. It has also consequences for quarantine. You feel safer simply because you said, oh, you had this cough, you had these symptoms without knowing it was the corona infection. We had cases in January here in Germany. We had them in February. Waves, little hotspots, but this is not surprising nowadays. You know, the discussion in France, the first case, they suspect in December now. So, but I'm sure we had people traveling to China, professors, I know they came sick back in January here to Munich, and these are proven, and we found antibodies in these people.
Okay, thank you very, very much. I'll just ask a few of the questions that have come in. Leona, I think this is directed towards you about this test specifically. I do think you've probably answered it as well, which is, if you stop sharing, Armin, then we'll be able to see your faces larger again.
Yes, give me a second.
I'll just continue asking the question, Leona, which is, can this be used in an emerging infection or only after 7 days?
Well, the literature right now, as Dr. Schwarzbach mentioned, the antibodies start being produced at day 3, and they show immensely at day 7 for IgM and can peak even later on, and then IgG takes over. So we have to wait the 3 to 7 days anyways for IgM. Yeah, Armin's showing it again here. So this is actually quite good. So this, in the beginning, you can see that.
Armin, if you go, is this the one you want, Leona, or the one before that?
Fine. No, this is okay. This is traditional textbook response of how antibodies, are developed in any type of kind of situation where there's foreign microbes coming in. So IgM spikes up usually anywhere from 3 to 7 days, and then IgG. In this case, it's showing IgA, which is quite nice as well, showing up. And so we just have to wait until biology kicks in, until our immune system kicks in and starts producing the antibodies.
Okay, thank you. And then this one, I suspect you may already have partly answered. I'll just read it as it is here. Do you think that herpes viruses have any effects on the test results or on the chances of succumbing to this virus?
Yeah, so again, there's 2 questions in here. I think that all these test kits have to check the robustness and the cross-reactivity against different viruses. And we tried to use as many as we had, like in our biobank, and as well as donated samples. So for us, herpes did not have underlying cross-reactivity. It's not shown here because we didn't have the numbers to actually give kind of solid data. It's one of those patient groups that we need more samples to test. But I wouldn't be actually too concerned with it. It's been reported from the previous SARS cases in 2008 as well as in 2002 that herpes virus did not have such a huge effect on this performance and robustness of the test kits. And then the other question was, what are the chances of succumbing?
Or on the chances of succumbing, yes.
Yeah.
To the virus.
Well, I think, and again, I think that the research right now in regards to those high-risk individuals are those people that have underlying comorbidities. And if that is then a herpes infection, then of course that puts these individuals at a higher risk. So anything that actually affects the immune system is going to put individuals at high risk. And these comorbidities, like a herpes infection, like a CMV infection, like a human parvovirus infection, will put individuals at a higher risk. And that's just, again, been reported in the research, even especially for this virus as well.
Thank you. Martin, could you just help Armin to stop sharing his screen? Because I think, Armin, that's what you're trying to do.
Yes, it's a little problematic. I try to stop this. I need help.
You need the bottom, Armin, at the bottom, middle. Put your cursor down there, share screen. Click on that.
And stop.
Do you want to continue? No.
Well, this picture is quite fine. The antibody.
So now
This picture is fine. It's just a pity not to see their faces bigger.
good now.
Do you want to stop others? You want to continue? I think it's yes here, Armin. If not, it's no.
I'll just go on with the next question, Leona, which is, could this virus lurk in the body like herpes? Do you think it could? Do we have any evidence of that yet?
We don't have any evidence yet of this one. We don't. And so in looking in the body means like integration into our genome, or actually, you know, just kind of evading the immune system. We don't have the signs yet to indicate that. And actually, I did check from the previous 2002 incidents in 2008, and the science is not strong enough to suggest that it does. So we just need more research in this area, and I hope that there are signs. I know that there are scientists looking into this. Yes.
Okay. Well, if the infection shows positive 6 weeks after the person has actually become infected, does this mean that it can still be passed on after 6 weeks?
What a fantastic question.
Mm-hmm.
Because we are in self-isolation, aren't we? We are socially distancing ourselves to flatten the curve. And, oops, okay. So the incubation period or the risk period apparently is anywhere from, as Dr. Schwarzbach mentioned, anywhere from 2 weeks to 4 weeks, okay? So 6 weeks is obviously after that. So unless we intermingle with high-risk individuals, like primary healthcare providers, or those individuals seeing other infected areas, individuals, I do not think we're going to be seeing new cases. If you look at New Zealand as an example, they say they have stopped the cases, completely stopped it, and that's because of the social isolation, and that's because of actually taking precautions of not actually intermixing those high-risk individuals with low-risk individuals. So this 6 weeks is enough time for this virus. If you had it, you will succumb to its symptoms or not, but you will get over this being asymptomatic and just get over the infection if you have been asymptomatic. Your antibodies should show up by then, of course. As Dr. Schwarzbach mentioned, antibodies can stay in your system, as he gave you that lovely diagram, even after 240 days. So 6 weeks is really past that. I hope I answered the question.
Well, thank you very much. And then there's another question, might be quite complex. Is there an immune response pattern between dengue fever and or hemorrhagic dengue and COVID-19? Is there any science in on that yet?
Oh, I haven't seen it.
No,
I haven't seen it. But I know that FDA wants the test providers to actually try to do a validation study with the different species of dengue and their tests. So the point is, with doing these validation studies, we need to have samples and positive samples. So obviously a negatives characterized sample. So I think that would be the bottleneck to actually do this type of validation. But let's see what kind of samples we can get from these reference companies.
Okay, thank you. Another question that was asked before the seminar or the webinar, Armin, was whether any of the tests that you carry are yet being used in Germany as a kind of health passport. In other words, I think you've already answered that. It's not possible yet, but it's what people are hoping, is that if they have an IgG, they'll be able to utilize that to sort of, prove that they've had it, but they don't have it anymore. Is there any sign that that's coming through in Germany as a sort of an option, or is it just too early for the science still?
Our health minister is discussed that already to start a discussion. Yes, we have some tendencies, but the ethical committee in Germany needs to check this. If it's we have a discussion about that, like vaccination discussion. Can we urge people to get vaccinated? Could we put pressure on them? This is a high discussion, but what is clear, we have in every vaccination, we have a passport. If you do, as an example, tick-borne encephalitis vaccination or varicella zoster, whatever, or measles, you get vaccinated and then you have a passport for it. The question we need to answer then, how high needs to be your titer and how long is your protection? And we don't have any data about that. We will get it maybe in 10 years or so, or 5 years.
But it is accumulating gradually, isn't it? Dr. Schwarzbach, another question for you is what you think about the neurological or psychiatric sequelae of COVID-19.
Yes, we have also now a paper came also today about that, neuropsychiatric symptoms, and we know that it's neurovirulent for sure, like a lot of viruses are doing that, and they damage parts of the brain and the limbic system, and so we can get also neuropsychiatric symptoms. And what is now the prognosis in Germany, that we have more suicides. This is where we have now more panic attacks. We have now in Germany more depressions. I talked today with a psychiatric specialist about that, and she said, yes, we have now younger people, they get confused, they get panicking, they get depressed. So we have surely a lot of cases by that, and also damaged people, and also death cases by that, for sure.
No, really?
Yes, yeah. There's
That's
also
terrible.
prognosis nowadays about that, yes.
When you say death cases, do you mean suicide?
Yes, people are completely helpless. They lose their working places or whatever. So,
of course, not related to the illness itself.
Yes, not just this, the pressure, they get depressed. You know, we have a lot of people who have depressions in Germany. And what happens with them if they cannot go outside? The older people are getting completely crazy. I don't know how your situation in England.
Yeah, absolutely true
similar
here as well. Yes, no, that's definitely true. I'm not sure to what extent that's related to the infection itself, but it's more the political ramifications.
It's the consequences of doing us in a huge human zoo. We are all in a zoo. Yeah. And we humans are not used to that. Okay, wild animals, also you should not, monkeys you should not put into the zoo. That's the same situation. You restrict them, and there's a mental, and the old Romans, they said mens sana in corpore sano, so that means healthy spirit in a healthy body. And if something is going wrong with your spirit, you have a problem with your body. If something is wrong with your body, you have a problem with your spirit.
Yeah, no, it's in England something we're noticing as well, as nobody's talking about raising your immunity very much. It's just all about fighting the enemy.
Yes.
It's something we naturopaths and nutritional therapists are finding particularly difficult to cope with. But could I just thank you very, very much, both of you, because we're reaching the end of the hour and I don't want to extend beyond that. I know for you, particularly Leona, it's very late because you're 2 hours ahead of us, but maybe questions are still continuing to come in, so maybe we can have you on again quite soon, both of you, and plus further science will have come in another few weeks. Absolutely. So you'll be able to give us an update, just because it was asked on the in the questions where you can access this test, which we've heard about today, SARSPLEX. Please just call the helpline. That's Academy of Nutritional Medicine, AONM.org. And those on the helpline will be able to give you information immediately. It is a blood draw for SARSPLEX. So there are clinics open both in London and across the country. It isn't impossible to get blood draws. It is considered, you know, necessary if it's a health blood draw, but of course that has to be decided between you and your doctor or therapist to what extent you do that. Armin does have also other types of tests, for example, little kits that can be sent out to therapists and doctors, which enable you to do a test with your patient there and then, where you get the results within 10 minutes. That is an IgG test, isn't it, Armin? And I believe you're getting a lot of requests for those, because also in Germany people aren't able to travel that much yet. Is that true? You've had a lot of those recently done. Yeah, yeah, okay.
We
Well,
do a
we'll
lot of testings and for that, and a new development we established now is also for SARSPLEX. We can do it from a capillary system from the finger, so it works
really?
with that. Yes,
okay.
That is a new validation we did this week, because we have a lot of babies and children. They want to know, and you cannot put a needle into the vein, so it works with that. But we have a timeline of very fast doing it. It should be the capillary system. We have them, they are certified systems for babies or younger ones, so we can also test them now by that.
Well, that's fascinating. Well, we'll have to talk more about that, Armin, find out more, and then put out news on how that can be offered as well. So thank you both very, very much indeed.
You're welcome.
It's wonderful to have you both. Very, very interesting information, and look forward to hearing from you again. We will, by the way, be having further seminars in this series, or I should say webinars. We have Dr. Sarah Myhill next week on ME Awareness Day, and that's on Thursday, the 12th of May. She'll be talking about the whole panoply of chronic disease and how it's being affected by COVID-19. And we'll be having after that Dr. Robert Bransfield from the States giving us a webinar on more the neuropsychiatric implications of this. We have Craig Shimazaki, Dr. Shimazaki from the States, talking about, again, the interactions between PANS pandas and COVID-19. As you said, Armin, a big paper has just come out about that today, very significant. And we also have Dr. Horowitz planned as well for the, I think, 12th of June. So we'll be putting out a full list of all of those webinars, and hopefully at some point before all of those we will have come out of this lockdown, but let's just keep hoping. Thank you very much indeed.
Thank you, guys. Be safe.
Bye-bye, guys.
Bye. Thank you.
Bye.
Bye.